of MS, at least in terms of the autoimmune aspects. We have orally active beta modulators, which do certainly improve the quality of life for the patient, but in spite of this, what you find in MS lesions are the pool of oligodendrocyte precursor cells. These are the progenitors which form the myelinating oligodendrocytes. And in these plaques in the MS patients, these cells essentially become stuck, so they don't progress to cells which make myelin. And also the pool tends to be somewhat dysfunctional. So in spite of using targeting immunomodulation, the problem of demyelination and repair remains. So because of that, it's a very active area of research at present. And what people are looking at to enhance remyelination, two strategies: cell-induced therapy and drug-induced therapy. This slide just shows you some of the things that people are doing, working with induced pluripotent stem cells of human origin, rodent origin, fetal human stem cells, fibroblasts. And the idea is to either use growth factors or agents in terms of genetic modification to induce these cells to become oligodendrocyte precursor cells, which hopefully will form mature myelin-producing oligodendrocytes.
Okay.
And some of these are already in the clinic. The other route people follow is pharmacological, using drugs to induce progenitors or precursors to become mature myelin-producing cells. And a classical example of this, I think the first paper to come out was using benztropine. This is a muscarinic antagonist. It's approved by the FDA for Parkinson's disease. It promotes maturation of oligodendrocytes in culture. It improves outcome in animal model of MS, which is an experimental autoimmune encephalomyelitis, but it does have side effects, perhaps not the best thing you want to give to an MS patient. Now, the process of maturation of these precursor cells to mature myelin-producing oligodendrocytes is well characterized both morphologically and in terms of the markers which they express. So the precursors in vivo see factors like platelet-derived growth factor, which is mitogenic. So these cells at this stage express high levels of the PTGF receptor. As they progress, they lose this expression and express markers of the mature phenotype, myelin-basic protein, proteolipid protein, and other components of myelin. So you can actually use this to follow agents which promote this maturation pathway. Now, if you look at a mature oligodendrocyte in culture, these don't produce myelin unless they're in contact with axons, but they still express the building blocks of myelin. This one is stained for myelin-basic protein. What they do is they elaborate this rather robust network of fibers, which they would do in vivo, and this is what they use to wrap around or sheath axon segments in producing myelin. Now, one oligodendrocyte will ensheath somewhere around 50 axon segments. So you can imagine that losing even small numbers of oligodendrocytes can have rather dramatic effect on myelination and demyelination. Now, the idea of promoting remyelination as a repair strategy in MS goes back some time. Already in this review in 2002, the authors were proposing that what we should focus on is enhancing the long-term survival of the precursor cell and encouraging them to proliferate and also mature into oligos which produce myelin and remyelinate the sheath axons. Something also which we are interested in. Now, how can you go about doing this? If I just go back 2 slides, what you want to do is look at a pharmacological strategy which both enhances this progression pathway and also might promote the survival and proliferation of the precursor cell. So what we did for this was to put into culture, isolate oligodendrocyte precursor cells and put them under two different conditions. One, which we call an expansion medium on the left, a very simple medium which has growth factors, basic FGF, fibroblast growth factor, and platelet-derived growth factor alpha, which the cells would normally see in vivo. They have high levels of receptors for these factors. The other condition promotes maturation. A richer medium with hormone supplements and thyroid hormones, T3 in particular, is essential for production of oligos which produce myelin. So we started with the maturation condition and what we're going to study. We have a long-standing interest in molecules which are called fatty acid amides. A family of these are the N-acyl ethanolamines. Basically, it's a fatty acid hooked up to ethanolamine. A classic example is anandamide, in which the fatty acid is arachidonic acid. Anandamide is an endocannabinoid. It's an endogenous agonist of cannabinoid receptors. Other members of this family have, as the fatty acid, stearic acid, lake acid, or palmitic acid. These other molecules with the saturated fatty acid don't bind, don't activate cannabinoid receptors, but they do act as signaling molecules. They act as ligands for nuclear receptors, the PPARs, which are peroxisome activated receptors. Ligands for these receptors are anti-inflammatory and protective. They activate ion channels, so they do act as signaling molecules. They are found that are persistent. An interesting feature is they are produced on demand by cells when cells are exposed to stress or injury, suggesting that these molecules are part of an endogenous defense mechanism to promote or restore homeostasis when the cell is faced with stress. Now, the one we focused on is the palmitoyl ethanolamine, which has a long history of being anti-inflammatory, analgesic, and neuroprotective. So there is nothing in literature which indicates that it might have an effect on maturation of oligodendrocytes. So we were going to start these experiments when other studies came out describing a variation of this, in which the authors prepared a composite of this PEA, the flavonoid luteolin, which itself has antioxidant and anti-inflammatory effects. So they prepared this composite using what is called micronization. If you look at PEA under the scanning electron microscope, it's very chunky, it's very heterogeneous. And what is often done in the pharma industry when you prepare tablets of a drug is to use jet milling. So it's a grinding process to produce products which are homogeneous. So they did this to make a composite of the PEA and luteolin, and they found that was more effective than the PEA itself. A study done in spinal cord injury, the authors looked at the motor score in which animals were given this PEA luteolin composite or these single molecules alone and added together. So we thought, well, let's try this composite of PEA luteolin on the cultures of the oligodendrocyte precursors. So put them in the culture in differentiation conditions, added this composite, and looked at them under the microscope over a period of days, and were surprised and didn't expect this. See, after 4 days, the treated cells had a far more robust outgrowth of processes compared to the untreated ones. You see a small increase in the total protein content of these cultures, which is probably not proliferation, it's just increased tissue mass. If you look at Ki67, it's a well-established marker of proliferation. The gene does not change, this is in 4 days, in the cells expressed to the paleoleuthylene, so it's probably not a proliferation effect. They express receptors for CB1 and CB2 cameloid. This doesn't change also upon treatment with the paleoleuthylene. If you look at the PTGF-alpha receptor, as I mentioned, in vivo, in the precursor, it's very high because they're in a proliferation state. When you take them out of the tissue, put them into culture under differentiation conditions, this marker drops off very rapidly because they begin to differentiate. And after several days, it just falls off the table. It's not changed by the treatment, so that has no effect. So we asked what happens if we look at genes that are involved in components of myelin, like myelin-basic protein. Protein-lipid protein and myelin-basic protein are important structural components of myelin. They're expressed by mature oligodendrocytes, whether or not the cells are actually producing myelin. So they have the building blocks ready to go when the cells start to myelinate. This enzyme, long name, CNPase, is also upregulated when the precursors mature. And it's probably also important in the process outgrowth of the oligodendrocytes. So if you do a time course over 8 days of the precursors put into culture under differentiation conditions, you see that regulation of the genes for MBP and PLP without treatment. That's not surprising. They are under differentiation conditions and they do mature on their own. But if you treat them with the paleoleuthylene composite, the effect is enhanced. You see it both 4 and 8 days. As an example, we chose myelin-basic protein. You can see that the product of the gene, myelin-basic protein itself, which has 2 isoforms, in Western blot is upregulated. So the change in gene expression is manifest in the actual protein expression. Now, this complex, the TCF7L2, is a transcription factor which regulates the expression of myelin genes. It's upregulated when oligodendrocytes undergo maturation, both under normal conditions and also in the process of remyelination in pathology. The gene does not change over the 4 days in culture, but interestingly, treatment with the paleoleuthylene does lead to rather marked upregulation in its expression. It's actually considered a potentially druggable candidate for promoting remyelination. Now, myelin is composed not only of protein but also of lipid. Actually, it's about 80% lipid and much less protein. So we looked also at the effect of the paleoleuthylene treatment on the oligodendrocyte precursors under differentiation conditions. UTP glucosyltransferase 8, UGT8, catalyzes the final step in the synthesis of cerebrosides. And cerebrosides are an important component of myelin membranes. And this is upregulated during differentiation of oligodendrocytes. If you look at this marker in culture, again, with time it increases on its own, which is an index of differentiation. The cells are under conditions which promote differentiation, but the effect is enhanced when we include the paleoleuthylene composite. If you look at genes that are involved in cholesterol synthesis, cholesterol is also an important component of myelin. HMG-CoA reductase on the right, and this long one, isopentenyl diphosphate delta 1 isomerase, IDI1, over the 8-day time course, they are upregulated throughout the 8 days when treated with the paleoleuthylene composite. Interestingly, under control conditions, these genes did not change. But myelination is a very complex process. It doesn't occur all at once. It involves a coordinated series of events. So perhaps it's not surprising that expression of these genes for cholesterol, for cerebrosides, for myelin proteins, don't occur all at once. They have their own timings. Oxidative stress. We all know that in terms of energy consumption, the brain consumes far more energy in terms of its mass compared to the rest of the body. It's disproportionate. The brain makes up about 2% of the body's weight, but gives about 20% of the energy. Now, the highest rate of metabolic activity in the brain, we think, is not the neuron, it's the oligodendrocyte, because the process of myelin production is very energy intensive. In fact, in oligodendrocytes, when they are myelinating, the ATP levels shoot way up. Now, ATP production through the Krebs cycle and glycogen transport chain is what is called coupled. So, well, it is coupled. Some electrons do escape, so there is some production of free radicals during production of ATP. Now, the cells have a defensive mechanism for dealing with radical production. For example, glutathione scavenges radicals. But cells like neurons and also oligodendrocytes have a very low antioxidant protective baggage, very low levels of GSH. Another factor one needs to consider is that the brain's predominant iron-containing cell, not well known, is the oligodendrocyte. Now, iron is essential for production of myelin, but if you take a cell which has a high iron content, like the oligodendrocyte, very high metabolic activity, and a very low antioxidant defense capability, that's a recipe for disaster. So we asked what happens if we treat mature oligodendrocytes, 6 days in culture, with the paleolithium composite. Looked at two important enzymes for protection against oxidative stress. Superoxide dismutase, which breaks down superoxide anion to peroxide, and catalase, which breaks down peroxide to water. There was an upregulation of catalase by the paleolithium, not by the SOD, which is interesting because this is, as I say, an important defensive mechanism. And catalase has a very high turnover rate, incredibly high turnover rate. It will break down something like 5 to 6 million peroxide molecules per second. So perhaps also, apart from enhancing the differentiation, this treatment with the paleolithium may also enhance the cell's defensive capability against oxidative stress. Now, how does this work? Well, PEGA is, as I mentioned, it's an endogenous ligand for nuclear receptors, PPAR alpha. PPAR alpha knockout mice have no defects in myelin anyone has ever seen, or hypomyelination. However, mTOR, mechanistic target rapamycin, is a serine 3D protein kinase. It's a whole lot of things in cells. It's all the proliferation, motility, it's also survival protein synthesis, and essential for myelination. mTOR knockout mice have defects in myelination. Hypomyelate, oligodendrocytes don't mature. So we took our cultures, put them under differentiation conditions, looked at two representative genes, one basic protein and UGT8. Again, the increase of 4 days expression by the paleolithium, which was blocked in the presence of rapamycin, which is a potent and selective inhibitor of mTOR. If you block mTOR, the paleolithium does not work. There is a slight tendency to overcome the defect, but it really is blocked. Its treatment with the rapamycin also inhibits the morphological differentiation of the cultures. Does paleolithium activate mTOR? We don't know. We tried doing this looking at phosphorylation. The experiments weren't conclusive. The antibody wasn't great. The signal wasn't low. So certainly mTOR is involved with this direct effect or indirect of the paleolithium treatment. We still don't know. So the next set, the last set of slides, going back to this diagram. So we looked at the progression pathway of maturation. What about the precursor cells? Can we look at the effects of paleolithium on them? So what we did is to put them in, as I mentioned, a medium which does not permit their differentiation. So presence of growth factors FGF and PDGF. And if we look at the two media, this one look at differentiation versus proliferation. The PDGF receptor goes way down. The Ki-67 marker of proliferation, very high under differentiation conditions, very high proliferation, very low differentiation, which drops off. If you look at myelin basic protein in its cultures, at 4 days, in the proliferation medium is much lower than in the differentiation medium. So it tells us that under these conditions, the cells do express markers of proliferation and not differentiation. So we can indeed isolate two populations. So if we did add paleolithically to the cells under proliferation conditions, what happens at 4 days? These are controls on the left, treated on the right. So there are a lot more cells. They also appear, the precursor cells tend to be bipolar in morphology. These are more complex, interestingly, not like under differentiation conditions, but certainly they appear to be a bit more mature. This effect you can quantify, so there are more cells definitely in the presence of the paleolithium. We don't think it's a proliferation effect of the paleolithium. The cells are already in the presence of mitogens, so it probably, we think, enhances the vitality, and they respond better to the mitogens, which is interesting on its own right. Curious observation, if you put the cells under the two conditions, which favor either proliferation or differentiation, again, under differentiation, the paleolithium at 4 days increases the expression of MPP, which is blocked by mTOR. There was also upregulation of MPP gene expression when the cells were under medium, which promotes proliferation, which we did not expect. So it appears that even though they're under proliferation conditions, there is a tendency for this treatment, so encouraging to along the differentiation pathway. The next to last slide, is what happens if you take, this is a model of what is called experimental encephalomyelitis. You treat mice with a fragment of myelin oligodendrocyte like a protein. It's a myelin structural protein. And this immunization induces an inflammatory response, much like in MS. The animals undergo demyelination and they become paralyzed, which you can measure with a clinical score. So, and the signs appear after about 11 days post-immunization. And if we start treatment at 11 days with the paleolithium, and you can see that at all the time points there was a significant reduction in the clinical score. The effect is probably underestimated because we see, this is the composite of 3 experiments. We treat the animals with the immunogen without the paleolithium. One or more of them become extremely ill, they have to be sacrificed or they die. And these animals are excluded from these calculations. So the effect is probably underestimated. So what we need to do now is look at the actual histology if the myelination is being preserved in these animals. So the last slide, conclusions. The co-altered paleolithium treatment promotes the morphological and biochemical differentiation of oligodendrocyte precursor cells, favors the survival of the progenitors, and improves clinical score in the mouse model of EAE. So we think this may represent perhaps a novel pharmacological tool to promote myelin repair in such diseases. So we'll await the studies so I think my time is perhaps really out, so at that point I will stop and take any questions if there are any. Thank you.
Any questions and comments to this presentation? No questions. It means everything is very clear. Thank you very much.